Dear students, as soon as the telophase is about to complete or at the later stages of the telophase, what happens is that the astral microtubules they will send signals to the equatorial regions or to the plate regions of the cell, and there the actin and myosin they become activated, and then they form contractile ring, which will form the constriction of the cell in the form of a furrow. Furrow means a constriction, the cleavage furrow. Cleavage means it will divide, and furrow means a depth, like this. These are the newly formed nuclei with chromosomes. Now here, like this, movement will be in this direction, like this, the cell will be divided. This cleavage furrow, this is cleavage furrow, and it will deepen until the cell is divided into daughter cells. Now the mitotic events in the plant cell, let us discuss them. Mitosis of plants. Where the differences lie. First of all, the plant cells they do not have any centrioles. The basic events are same the prophase, the metaphase, anaphase, telophase, but plant cells do not have centrioles. So how the mitotic apparatus will be formed? They have the analogous or similar region. The analogous region is just like centriole, and it will form the spindle microtubules, or you can say the spindle fibers. The second difference, so this was the first difference, that there is no centriole. Now the second difference is that the shape of plant cell will not change. As we observed that during the mitosis, the cell from round it will become elongate, and finally it will be fully elongated or stretched, and then it will be divided into two cells. But shape of plant cell remains same. When we compare it to the animal cell. Now what is the reason that shape remains same? This is because plant cell is surrounded by cell wall, and cell wall is very rigid. Because of that, the shape of the cell remains same during the process of cell division. The third difference is that at the point of cytokinesis, now at the point of cytokinesis in plants, in animals, we observed that there is formation of cleavage furrow. Now let us see what happens in case of a plant cell. Now this is the cytoplasm, and here two nuclei have been formed with chromosomes. Now what happens, that a structure phragmoplast will be formed. What happens that the Golgi bodies or the Golgi apparatus will send, or they will form some vesicles The vesicles at the point of metaphase, they will arrange at the center. Then finally they will fuse with one another, or they will merge with one another, and they will form a disc-like structure which is known as phlegmoplast. Now this phlegmoplast will divide the cell. The membranes of the vesicle, they will become the plasma membrane. Now after the division, this phlegmoplast will help in the formation of cell membrane. The vesicles also contain the material for future cell wall, such as it also, now the Golgi bodies are sending vesicles. The vesicles have the material to form the cell membrane and cell wall as well. There are some amounts of cellulose and pectin, and cellulose and pectin are important components of cell wall of plants. So this is the basic difference between the cell division of animals and the cell division of plants, that first they have no centriole, the spindle fiber will form from an analogous region, then the shape of the plant cell will remain constant throughout the cell division, it will not be disturbed due to the presence of cell wall. And the third one is the cytokinesis. Here the cleavage furrow is not formed, instead the formation of phlegmoplast from the fusion of vesicles that originate from Golgi bodies, and they contain the precursors of cell wall and cell membrane. Dear students, that was all about mitosis, the process of mitosis. In the next video we will be discussing about importance of mitosis and how the abnormality in any mitotic function or mitotic division will result in an abnormality in your body. That will be discussed in the next video. Till then, stay tuned, keep watching office.com.pk. Thank you so much. Allah Hafiz.